economic resources to remove them. The next challenge is in terms of the scalability issues. Successfully developing and optimizing processes at the laboratory, as we all know, does not mean that the effort will be easily replicated or achieved in the industry when we want to scale up. The final challenge in sustainable bioindustry is the competition from fossil-based products. This competition is real because of the fact that fossil-based alternatives are actually much cheaper than the bio-based products. Next. Next slide, please. So in order to circumvent or a strategy to overcome these challenges, we can actually utilize multi-omics platforms in order for us to integrate data so that we can understand the processes that occur in biological systems at the holistic level. And omics platforms such as genomics will give us basic information on all the genes contained within the organism. And then transcriptomics data will give us information on specific conditions of that particular plants or microbes. And then proteomics will give us information on the enzymes involved. And I think when we're talking about bioindustry, the main omics platform normally utilized is the metabolomics, which gives us all the information on all the compounds or secondary metabolites produced, which are of interest. And of course, there are other data such as epigenomics or even so-called phenomics, where we can have information on those specific aspects. This information on the multi-omics platforms allow us to understand how plants grow in different ecological locations. For example, the influence of regional areas or altitudes on the plant development of plants, as well as stresses, biotic and abiotic stresses, how they influence on the important traits for crops such as rice, maize, peanuts, potatoes, sugarcane, and rapeseed. Next slide, please. Next slide. Okay, so information from the omics are expected to allow us to have the so-called data-driven design, and we need to optimize them in order for us to be able to identify the metabolic bottlenecks. These multi-omics data can point out to us the specific steps that occur in the metabolic pathway, the regulatory steps, so that any of the blockage or the bottlenecks in the increase the level of the production. So the downstream process is that synthetic biologists can actually utilize understanding about the pathway in order to remove the bottlenecks and increase the production. Second outcome of this data-driven strategy is to be able to predict biological outcomes. So by analyzing this multi-omics data set, of course, that requires other fields such as using machine learning and AI. We can produce predictive models of how the engineered microbes in the downstream synthetic biology can be stimulated, how we can design them to increase production. So this will enable the so-called rational design of the strains. And the omics data can allow us to understand the complex interaction. It is not sufficient to just utilize a single omic layer to understand the complex microbial system. So we need to integrate all of the omics data to understand the intricate connections between genes and phenotypes so that we have a clear overview about the whole organism or the, the cellular mechanism at a holistic view of the entire biological system. Next slide, please. So I want to share with you the strategy that we have used in understanding about production of egg wood. And I think many of us are aware about the importance of egg woods in our everyday life. And for your information, the egg woods market share by the region has been postulated for the period of 2021 to 2033. And for last year, 2024, the market share value is estimated at 145.2 million US dollars, with North America contributing the most amount at 40%. Asia Pacific contributed 23%, whereas the projection of the production in the world has been estimated for 2031, with cumulative annual growth rate of 3.2% for North America and 7% for Asia Pacific, which means that Asia Pacific is expected to make significant gains in terms of the market share. And these are mainly due to the rising consumer demands for three aspects: for luxury fragrances, traditional medicine, and wellness products. So when we talk about traditional medicine, the egg wood is actually utilized has been utilized to treat common ailments and are commonly used by especially the Asia Pacific region. And this is due to the metabolites produced by egg wood. Next slide, please. Now I want to introduce to you the species that one of the species or one of the genus that produced egg wood, which is Ecloria species. These are large evergreen trees belonging to the Thymilaceae family, and they're found in the rainforest areas in Asia. There are actually 31 Ecloria species worldwide, but only 19 produce egg wood. And the importance of egg wood, as I mentioned just now, is the fact that the egg wood oil is an important ingredient in fragrances. And for aromatherapy, for incenses and aromatherapy, as well as traditional medicine, there are at least 31 luxury brand perfumes that use egg wood oil to produce their perfumes, and these are some of the names that produce them. In Malaysia, the species of Ecloria that are popular Equilaria melicensis, Equilaria beckeriana, Equilaria hirta, Equilaria rostata, Equilaria microcarpa, and Equilaria sinensis. The primary producer of egg wood in Malaysia is Equilaria melicensis, whereas the other species that is of interest and gaining and is also important in Malaysia in terms of economic value is Equilaria sinensis, which is mainly imported from China. Equilaria species grow on flat lowlands, hillsides, and ridges at the level of below 300 meters above sea level. And in Malaysia, there are approximately 2,000 hectare plantations with 2.3 million trees. Next slide, please. So how is egg wood formed? Egg wood is believed to be the product of a plant defense response. And this happens when the trees are injured, and the injury or the wound causes the production of signals to the rest of the tree, and finally allow egg wood to be formed. There are two categories of injuries. Naturally, the egg wood is produced through the induction as a result of physical damage or wounding, such as broken branches or wounding caused by insect attack, which are actually followed by microbial infections. And this kind of this principle of agarwood formation, natural agarwood formation, is mimicked by the artificial induction, where the injury is caused by physical injury, followed by chemical stimulation or microbial consortium. And as a result, agarwood can be formed. The infection by microbes uh, actually lead to the formation of the structure called tylose. Tyloses are formed within the xylem vessels upon wounding. These are overgrowths of parenchyma cells which protrude into the xylem vessels through the pits. And the wounding is actually followed by the release of gums, resins, and waxes as defense materials, which will cause blockage to be formed in the infected area and prevent spread of the infection. Uh, Together with this formation of the blockage is the release of volatile compounds or secondary metabolites, such as terpenes, which produce, which are the important secondary metabolites uh, in the agarwood oil. Next slide, please. So the problem with the industry is that there is high demand for agarwood, and it actually results in the overzealous felling of trees, which leads to loss of natural habitat. As a result, the agarwood species, all of them, especially Agarwood americansis that we have in Malaysia, becomes endangered species listed under the Appendix 2 of CITES due to depletion of the natural resources. Another problem in this industry is the fact that the natural formation of agarwood is a very slow process that takes several years. And to fulfill the demands, the high demands of the industry, there are also increased occurrences of adulteration and substitution in agarwood trade, which means that very low quality agarwood or even infected or even mixed agarwood sources are being used in the industry. Next slide, please. So the solution for this problem is reforestation of coral trees to circumvent the loss of natural resources, as well as artificial induction. But the problem with artificial induction is that different methods actually will produce agarwood with different metabolic compositions, and they are found to affect the aroma and quality of the agarwood being formed. And if this is talking about production of agarwood oil for the usage in perfume production, but in fact, these metabolites produced in the metabolite composition produced in due to different methods will also affect the biological activities of agarwood. For example, what has been reported for antioxidant activity. Next slide, please. So the strategy is to use the different omics data and integrate them so that we can be informed about the choices and the strategies to overcome the limited resources of aquaria species in nature. Next slide, please. So for Echolaria species, I'm going to share with you the information that we have regarding Echolaria species. Of course, in our lab, there's many of the omics that are not done. I will share with you the resources available for Echolaria that have been done by researchers in this field. In terms of the genomics, there are actually only two information on the genomics data. One is on the first draft of the genome for the genome sequence of Echolaria aegoloca. Aegoloca is actually another name for Echolaria melicansis. So that this is the original resources for a genomics sequence. Okay, and it gives us information on all of the genes available in the plants, so that later on when we try to study transcriptomics, then we can utilize this gene sequence to annotate the transcriptomic data. The second genome data is available for the Equus sinensis. Of course, there is a lot of work that has been done on Equus sinensis, mainly because this is the species that's being worked on by China. They have very large community there, research community there, working on Equus sinensis, and mainly because this is the type of fragrance that they prefer to be produced in their incense. Next one. Next slide, please. Next one is on the proteomics. There's only one large data set on proteomics that has been reported. The rest is more, the other reports have only been on certain enzymes. This is the only one that actually look at the abundance of proteins or proteomes in Eclaria, which is the work by a group in Malaysia, in University Putra Malaysia. They got proteomic strata from the wounded Eclaria stems, and basically this study corroborated with the transcriptomic strata the fact that to show us about the terpene synthase pathway, which has been observed to be enriched in the proteomics studies. Next slide, please. So our approach is actually, we are only able to look at two aspects, and we feel that this is the most practical ones that we can contribute. We work with acromanicensis, and we actually utilize metabolomics and transcriptomics to understand about the agarose production in artificial, in natural, during natural formation, as well as due to artificial induction. Next slide, please. So for the transcriptomics, as we know, transcriptome gives us information on the RNA being produced in certain cells, in certain tissues, at very specific conditions. And with the information from the genome data, we are able to annotate transcriptomes for these two conditions, these two egg woods, the naturally formed egg wood as well as the egg wood formed through artificial induction. And we've got information on the terpene and phenylpropanide biosynthesis, which are the main metabolites produced by Eclaria. Next slide, please. So from the transcriptome data, we're able to look at, to distinguish the transcripts that are mainly produced in the naturally formed egg wood compared to the artificial induction. These are the numbers of transcripts. Next slide, please. Right, then the transcripts, the transcriptomic data will later on be integrated with the metabolomics work. So from metabolomics, we're able to look at the metabolic profile produced. And so far we've been using GCMS only, but then if we want to look at the details, LCMS of course is better, but because we are just looking at volatile compounds, GCMS is sufficient for what we do. And other groups have reported on the profiling of the chemical constituents specific for the egg wood that they study. We can actually use this profiling to identify the egg wood that we study, whether they are naturally formed or they're actually artificially induced. Next slide, please. We have actually found metabolites which are significantly found in naturally formed egg wood, and this can become the standards for us to evaluate any egg wood produced by the artificial induction to show the quality of the methods, the artificial induction methods used. Next slide, please. We compared the metabolite profile with the standard, with the control, which is the healthy tree without the egg wood being formed. And uh, go to the next slide. We will see the heat map of the metabolite profile. Next slide, please. Okay, this one here. So we found significantly produced metabolites, and we can group them. We find that they are some of them are highly highly produced in naturally formed egg wood, 67, compared to artificial induction, and then also some actually low in the level of the metabolites are lower in artificial induction compared to naturally formed egg wood. Next slide, please. So the main group of compounds produced are sesquiterpenes, and which are followed by monoterpenes. And we found that uh, for the egg wood, for both of them, naturally and artificially formed egg wood, sesquiterpenes are found in both of them. That means sesquiterpenes are only produced by egg woods. But then, in terms of the level of the egg woods being formed between the naturally formed egg wood and, sorry, level of terpenes produced in the naturally formed egg wood compared to the artificially induced egg wood, there are actually higher concentrations of these terpenes, sesquiterpenes, and monoterpenes in the naturally formed egg wood. The artificially induced egg woods also contains coumarins. So this means that if we receive an unknown egg wood and we study the metabolite, we can actually inform the user whether this egg wood is artificially produced or naturally formed. Okay, so uninduced wood actually contains a lot of aldehydes. Metabolite composition varied significantly between different induction methods, and we found 19 compounds which were to be significantly different. So from the next slide, please. From the transcriptomic data and the metabolite data, we actually mapped them onto the biosynthetic pathways of sesquiterpenes and monoterpenes. This is for sesquiterpene. There are 7 genes that we identified, as well as 9 metabolites, and they were mapped onto the pathway. Next slide, please, is on the monoterpenes, right? Now, may, many of the researchers in this field actually study sesquiterpenes more than they study monoterpenes, but the enzymes involved in sesquiterpene biosynthesis are actually very difficult to study because they produce multiple types of compounds. And if you want to produce single compounds, it's almost impossible if you're using sesquiterpene synthase. So we focus our work on production of monoterpene synthase because we can then use one monoterpene synthase to produce one particular monoterpene. Next slide, please. That is the study on the production of the terpenes in terms of what is contained within the egg wood oil. We also wanted to know about the effect of the different induction condition, how the egg wood is formed due to different inductions, how it influences the antioxidant activity. Many groups have actually studied antioxidant activity of Ecularia, and most of the work has been done on leaves. I think there's only one work that actually looked at the antioxidant activity of the egg wood itself. And the reason why we are studying it on the egg wood itself is because locally, the, the, the users are actually taking in or consuming not the extract itself, but uh, the hydrodistillate from hydrodistillation. They actually consume it, and they believe that it has this, it can actually cure diseases such as the usual ailments like diabetes or even cancer. So we wanted to know the antioxidant activity in the egg wood itself. And we found that naturally formed egg wood extract showed the highest antioxidant activity compared to artificially induced egg wood extract. And we were using different uh, chemicals to extract them, and ethyl acetate is found to significantly to show significant difference in terms of the different egg wood samples. But as a whole, we know as a conclusion that the antioxidant activities were influenced by different metabolite composition in the egg wood. So from this information, we know that it's important for us to understand to actually identify the source resources, the source of the egg wood itself, whether it's formed, naturally formed, or artificially induced. And but in terms of its medicinal activity, it doesn't matter whether the antioxidant, whether the egg wood is formed through naturally or artificially induced. Both of them will still have this antioxidant activity. So, what is the outcome of this? Next slide, please. Uh, how can we utilize this information to ensure sustainability? And then downstream process from this is, of course, synthetic biology. So in synthetic biology, microbial cell factories will be utilized to produce the microbes that can produce this artificially. And this allows the for us to engineer organisms to produce biodegradable plastic, for example, and bio-based textiles, so bio-based materials. And then this will allow us to uh, capture atmospheric carbon dioxide and convert it to usable chemicals. Of course, in biosensors development, this is the most advanced aspect of uh, synthetic biology. And finally, engineered microorganisms can be developed to enhance crop productivity, resistance to stress, and nutrient uptake. Next, so in our effort to utilize synthetic biology, we have actually um, uh, use this following the strategy that I'm going to share now. We actually introduce another plasmid that carries part of the terpene biosynthetic pathway, which is the last steps. If you look at the diagram, the plasmid, additional plasmid that we require is what's called the PDXS DXRIDI. It carries the three enzymes, which are the downstream enzymes that will allow production of the precursors for this terpene
Bu
synthetic
Dr.
pathway.
Haida, halo.
Yeah. Hello. Yes.
Halo. Dr. Haida, I'm sorry, but due to our time limited schedule, I would like to give you 1 more minute to finish the presentation. I'm sorry.
Okay. Yeah. It's okay. It's okay. We use this plasmid together with the plasmid that contains the gene sequences for the terpene biosynthesis. Next slide, please. So following the diagram here, this is how we actually introduce the plasmid. So we have the main plasmid that contains the prenyl transferase together with the terpene synthase, and then we co-transform them into the same E. coli, and then finally we can, because the precursors IPP and DPP are produced in vivo, then we can have production of the terpene in vivo. So using fermentation, we're able to produce. Can you go to the next slide, please? Okay. Using fermentation, we're able to produce the monoterpene at a level which are about more than 10 times higher than what has been reported. So, what is the combined impact of multi-omics approach and systems biology and synthetic biology is that the multi-omics produce the data we require, and then we can design the microbes to produce the metabolites of interest, and then metabolite then can be optimized, and finally, well, eventually, then the industry needs to be able to scale it up. So in conclusions, next slide, please. So we utilize multi-omics approaches to transform sustainable bioindustry in order to provide the information required for uh, the downstream synthetic biology to be able to produce specific compounds required by the industry. That's all. Can you go to the next slide? These are the references that I have used, some of the references. Next slide, please. I want to mention the acknowledgement. That's the most important part of the presentation. This work has been supported by the grants from the Ministry of Higher Education Malaysia, as well as University Kebangsaan Malaysia. The researchers in the group are from Institute of Systems Biology, Dr. Kamran Azlan Azizan, my students, Yashidi Simpasivam, in the Faculty of Science and Technology, my students, Farah Adiba Abakadi and Afiq Adham Abdrasi. The Eggwood work has been done together with company Wafika Plantation and Services. Thank you for your attention.
Thank you very much, Bu Rohaida. Please give a round of applause for Dr. Rohaida. Ibu, please do not leave the Zoom meeting because I would like to invite our next invited speaker to the session, and after that we will have the question and answering session. So we will probably do that in around 25 minutes. So I would like to welcome our next invited speaker in this session, Dr. Indrawan Cahyo Adilaksono. He received his doctoral degree in agricultural science from Kyoto University, and his research focuses on agricultural mechanization and hydroponic technology, and then the application of ultrafine bubble application in agriculture. So without further ado, I would like to welcome Dr. Indrawan.
Thank you very much, Bonofi, for the nice introductions. Okay, maybe first of all, I would like to briefly introduce myself. So I joined with the School of Life Science and Technology in 2019, and after that, I have the opportunity to continue my doctoral program in Kyoto University, and I just finished and graduated last year, 2024, and now I have the opportunity. So the background story is 2 months ago, I have the message from Dr. Taufikur Rahman to be one of the committee, but 1 week after that, I become the invited speaker. So yeah, this is an honor for me to give a short presentations. Yeah, I try to make it as quick as possible. So the title is the engineering hydroponic system for control agriculture. So basically, I would like to share my experiments about the plant factory and also ultrafine bubble technology applications in plant factory, and a bit about the result of my experiments. And please, next slide. Yeah, before we go deeper into the control plant production system, I would like to we to see the big picture what happened in the world right now. So from this figure is from our work in data. We can see that maybe next in 2023 there are 8 billion people. So means that it's a huge populations already in the world, and it's expected increase into 10 billions people in 2050. Next, I have this one right. Okay. Um, if we see the data, when we see the urban populations and also the agriculture populations, historically in the past, the people, most of the people is live in the rural area or agricultural area, but in the future, most of the people will move to the city, and the urban population will increase. The urbanization will expanding means that the land area will decrease for farming. They will be occupied by house, industry, or infrastructure. And in addition to that, the number of the farmer is decreasing year by year due to the aging and the young generations move to the city. And according to United Nations in 2017, uh, to feed 10 billion people in 2050, we need to increase our food productions capacity by 70%. And another important factor is about the increasing of the global average surface temperature. We can see in this graph that since 1980s, the temperature, the global average surface temperature is increasing by up to 1 degree Celsius. So the farmer suffer with the long drought seasons and also heat stress and also the crop failure. And also we can see nowadays we have a abnormal rainfall patterns, and it's caused it reduced the yield and the quality of the agriculture products. And to avoid that food crisis, the innovations to engineer the plant production system that respect to environmental economics and social sustainability are crucial. And maybe not all of you familiar with this terminology, the plant factory. Uh, there are two types of plant factory. Maybe in the Europe, it's called as the IVF or indoor vertical farming system. And in Japan, it's called as the plant factory. There are two types. The first type is this one, the plant factory with the artificial lights or PFEL. And the second type is the plant factory with the solar lights. And our group of research in the Kyoto University tried to construct a sustainable food production system with the power and resource saving through our research using plant factory facilities that we have in university. And this is, I skip. So this is the typical closed step plant factory in Japan. And I think this one is that we have in the Osaka Prefecture University. This is the research type 1. And I have visit one of the commercial plant factory in the Kameoka City, Kyoto. And almost the same. In general, it has the germinations area, this one, and also has the nursery area and also the growing area. This is the harvesting area and packaging, and there are some warehouse and work area. And plant factory itself is defined as a facility designed to assist grower in achieving consistent year-round productions of high-quality vegetables by artificially controlling the environmental conditions. So the main components of the plant factory, there are maybe 9 components like a lighting unit and so on. And the benefits of this technology is provide the stable crop productions and also maintain the high quality and the quantity of the products, and also provide the comfort working environment for the young farmer, because we know that I uh, give a lecture in the Department of Agriculture Engineering, and I think most of my students don't want to work in the agriculture sector. So it's important to have a comfort working environment for them, and also this facility can build in almost anywhere, yeah, even in the harsh area conditions. And yeah, environment, some parameter that usually we try to uh, explore is like lights, water, temperature, fertilizer, CO2, and airflow. Photo periods, light quality, light intensity, the directions, amount, and so on. And of course, there are some challenges about this, the application of the plant factory. The first one is about the limited type of crops that can be cultivated inside the plant factory. Um, So far as I know, they are just maybe vegetable, herbs, and maybe some of a flower medicine plant that can cultivate inside the plant factory. But like a staple food like rice, wheat, and so on, they cannot cultivate inside the plant factory. And the second problem is about the initial costs that have to spend to build these facilities is around 4,000 US dollar per meter square, and also the high production costs during the operations of this facility, uh, especially in the labor cost and the electricity. In Japan, most of the university try to uh, change the human with the robots. So there are some robots that develop to do the germinations, transplanting, and so on to reduce the labor costs. And there are some research also about how to reduce the electricity costs during the operations. There are some key point like using advanced LEDs technology, improving the lighting system, improving the light quality, optimally controlling the environmental factor, like I mentioned before, and improve the culture methods. Okay, and this is one of topics that uh, con, the research conducted in the university, like try to produce the crop efficiently using the artificial light with the wavelength effective for plant growth that Prof. Iriawati already mentions about the importance of the lights for the plant growth. We try to find the right intensity, right configurations, and so on in the plant factory. And also this year I have the fund for, I have the students who work with the LED. We built the LED grow lights in the School of Life Science and Technology. And the other topics about the automatic nutrient solution management system controls the concentrations and the acidity and alkalinity of the nutrient solutions. And in my research also I apply the aeration technology. Later I will describe is about application of the ultrafine bubble technology in the nutrient solutions. And we also monitoring CO2 concentrations for photosynthesis. Control the CO2 and temperature also important to make sure that we have a stable productions of the crops. And else, and the last is the application of ultravine bubble in hydroponic cultivation inside the plant factory. Actually, there are some already have some report about the effects of the plant. I mean that ultrafine bubbles on the plant growth, like promoting the growth, reductions of disease, and so what is the according to has the diameter size less than 1 microbubble. I'm sorry, 1 micrometer. Maybe you familiar with the ordinary bubble. It has a 1 millimeter in diameter, so it's visible and rise fast and burst at the water surface. But fine bubble is consists of two, micro and ultra fine bubble. Micro has a appearance like a milky and cloudy-like appearance. The diameter range is between 1 to 100 micrometer, and yeah, and it has better mass transfer compared to millimeter bubble, and ultrafine bubble is more smaller, less than 1 micrometer. And we cannot see it's invisible or transparent when in the water. So there are a lot of methods to generate the fine bubbles. One of them is the method is pressurized dissolutions method. So I make it simply. So we pump the water through this small gap of the pipe. Maybe it's we call this the venturi. So there is creating negative pressure in this area. So the air sucked into the pipe, and liquid and gas mix in that area. And after that, the pressure goes up again, and gas dissolves to the liquid under slightly high pressure, and then liquid exit at to atmospheric pressure become tiny bubbles. Okay. Uh, as far as I know, in 2010, the application of fine bubble is start. Usually, the application is for wastewater treatment, and after that, develop in aquaculture, promoting the fish growth, reducing the mortality rate. After that, for washing, for food, for medical care, and for in the agriculture, it has been reported for promoting the plant growth. Yeah, this is a bit about my experiments. So it's mentioned before that the application of ultrafine bubble can promote the growth of uh, the plants, but is as far as I know, is not the result is inconsistent. Means that sometimes it's good results, sometimes have a negative impact, or sometimes there is no effects at all for the plant. So. Uh, these two previous research says that positive effects, but it mentions that because of the high dissolved oxygens and the temperature is rising also during the cultivation and so on. So there is no research about the optimum application so far for UFP, and I want to know why it's inconsistent. So I want to investigate more deeply about the effects of a number of UFP for promoting plant growth. And this is the hydroponic setup, and the plant materials is freely isolated, and this is the cultivation stage and the growing conditions. And this is how I prepare the ultrafine bubble water. So in order to exclude the the effects of, and this is some, how to say, a treatment, like a pass number, how many times that circulated in and out to the UFP generator. And I also have a make a content index. It shows that how many uh, number of the bubble actually generate during the preparations. Yeah, this is some of treatments, and this is growth conditions in every stage. I harvest two times in the end of the nursery stage and the end of the cultivation stage. And I also measure the number of ultrafine bubble water in the solutions using the nano tracking analyzer equipment in Uji campus. And then uh, I measure the plant growth, the total leaf area, measure using the imaging. We also measure the superoxide accumulations using the nitro butyrazine test, and also conduct the TPPH test. And this is the result about so simply it says that when we generate for a longer time, we have a lot more UFP concentration in the solutions. And when we generate longer, it also mentions that the size of the ultrafine bubble is getting smaller and smaller. Yeah. And the result also shows it's a bit messy. I'm not sure why, but when we have a more ultrafine bubble number in the solutions, the we yeah more, but we need so means that uh, when we have a lot of ultrafine bubble number in the solutions, the plants uh, getting smaller in this area 100 pas, and the percentage of ultrafine bubble in the solution is smaller. That's shifting to more smaller uh, treatment. And I may I skip. So the reason is because uh, ultrafine bubble, when they stay in the solutions, they can collapse and produce the hydroxy radical, and it causes the oxidative stress for the plants. And it's also shown here that with the more ultrafine bubble treatments, the leaf area is getting darker, means that it receive more stress, oxidative stress the from the ultrafine bubble. So yeah, we generate ultrafine bubble in the solutions. It can remain for a long time. So free radical also generate when the microbubble is collapsed. It's produced the reactive oxygen species, which is already identified as a hydroxy radical. So it's stimulate the accumulation of endogenous ROS on the leaf lettuce, and in the moderate level, it can good for the plants. But if the ROS is beyond the plant threshold, it can be the oxidative. It can cause the oxidative damage for the plants. And maybe that's all for my presentation. Thank you very much.
Thank you very much, Pak Indrawan. Give a round of applause for Pak Indrawan. Okay, ladies and gentlemen, Bu Ruhaida. Okay, I'm just quick checking for if Dr. Ruhaida is still joining with us. Okay, while we are waiting for Dr. Ruhaida, I would like to open a chance for any audience in this session. If you have any question or a discussion that we would like to continue with either Dr. Ruhaida or Dr. Indrawan.